This is Module 4, Enzyme Reactions, Kinetics, and Inhibitors. This is one of my favorite subjects, guys, so let's get ready for this one. This is video 4-1, Introduction to Enzymes and Catalysis. Just because a reaction is favorable does not mean that it happens quickly. This is indeed true in our bodies. If we were to allow for glucose to be turned into or to be used to make ATP on its own without the use of enzymes, it would literally take thousands of years to process one molecule of glucose. We don't live that long, and clearly that's not what's going on. And so there must be something that's speeding up that reaction. Even though it would proceed spontaneously and on its own, it doesn't necessarily mean it's going to happen very fast. In reality, it does happen very fast. And so what is going on in our bodies? What is speeding up those chemical reactions? Is it the temperature? Because we know that you can increase temperature to speed up a spontaneous reaction or make a delta G more negative. You can increase the temperature to do that. However, we are living creatures, and if you increase the temperature of our bodies, we'll no longer be able to maintain homeostasis. Our cell membranes would fall apart. Our enzymes would unfold. Those structural proteins that hold together our cytoskeletons would fall apart. And so we can't increase the temperature to process glucose. So what else can we do? Well, we can add something that speeds up the reaction, but that does not directly participate as a substrate in the reaction. And so we're not adding another reactant. We're sort of adding a helper B, something that can help speed up the process without actually participating in the process. This has to be something that can be used over and over so that we can save on materials and energy, so that we don't just use these things so that they don't, so if we're using this extra substance, but it takes a lot of ATP to make the substance, so we keep using it up all the time, that would defeat the purpose. And so we need something that will speed up a reaction, something that can't participate in the reaction as a reactant, something that at the end of the reaction will not turn into something else like a product. It'll just remain the same. Something that we can use over and over, that we don't have to rebuild over and over. Enzymes satisfy all of those requirements. There are so many different types of enzymes in our body that catalyze different biochemical reactions. In fact, the far majority of biochemical reactions in our body use the help of an enzyme. Enzymes are classified based on the type of reaction they catalyze, and they usually end in ASE. Not always, but usually, ACE. And so if we are doing a redox reaction, those are called oxioreductases. They do oxidation reduction reactions. If we are doing some kind of isomerization reaction where we're changing one isomer to another, then we would use an isomerase to do that. An enzyme that creates a bond formed between two molecules could be possibly called a ligase. So in this class, towards the middle or end of the semester, when we talk about metabolism, we'll be talking a lot about isomerases, transferases, kinases, and phosphatases. Those are the four that are going to really come up a lot. Enzymes are highly specific for the ligand that they bind. Enzymes are globular proteins folded up to reveal what's called an active site, where this is where a substrate fits and binds, and it's very specific for the substrate they bind. The shape, the shape and the charge of the active site determine what can fit in there favorably. A substrate for an enzyme can also be referred to as a ligand, which is a molecule that can be bound to another protein, another molecule. So ligand is the general term for a substrate. When we're talking about enzymes, we usually don't refer to them as their ligand, but we refer to what they bind as their substrate. So that's specific to an enzyme. But a ligand can also refer to a hormone that binds to a receptor, or oxygen that binds to hemoglobin. The specificity of the interaction between the ligand, or the substrate, and its enzyme is based on its ability to fit in the active site properly. So it has to have geometric complementarity to the active site, like a jigsaw puzzle. And it also has to fit in there based on its charge. And so there has to be an electronegative complementarity too. If the substrate is positively charged, then the active site should be negatively charged for them to fit properly. The R groups of the amino acids, because they differ based on size and also based on charge, will usually contribute to the specificity between a substrate and the enzyme's active site. And so it's the R groups of the amino acids in its substrate that determine geometric and electronegative complementarity. This is sort of how, so this is related to the concept of stereospecificity. Remember, chirality matters, and the isomer that we're dealing with matters. The mirror image isomer of this molecule would not be able to fit into this binding site. And so the isomeric characteristic of the substrate is really important and determines its proper fit within the enzyme. And so not just the fit based on the shape, which is shown here by the little hump and the point here, fitting into the active site's enzyme, the enzyme's active site, but it's also the hydrophobic effect. And so there should be proper hydrogen bonding between the substrate and the enzyme. There also should be proper electronegative complementarity between negatively charged R groups and positively charged R groups. And so if this is a hydrophobic substrate, then the enzyme's active site should be largely hydrophobic. If it's a hydrophilic substrate, then the enzyme's active site should be largely hydrophilic. And then the shapes have to complement each other also. There are two models by which ligands bind to their enzymes, or by a substrate binding to an enzyme. There are two different types of models. There's the lock and key, which is the older model and has since been disproved. And there's the induced fit model, which is demonstrated here. So let's go through these two one by one. Here's the lock and key model, and this states that the shape of a ligand or substrate must exactly match the shape of the active site on the enzyme. So I'll stay here, sorry, I jumped ahead of myself. And so that means the substrate would be more like a key, and the enzyme would be like a lock, where there's only one key that fits that lock, and it has to fit perfectly. The wrong key will not fit in the lock, and so no reaction will proceed. So this is the lock and key model, very rigid, very strict. The induced fit model suggests that a loosely bound ligand that is not maybe a perfect fit, but is a near perfect fit, will interact with functional groups on the protein on the enzyme, and then cause a conformational change in the shape of the enzyme and the substrate so that both will fit more tightly. So that's the induced fit model. The lock and key model over here, the induced fit model over here. And so the induced fit model would allow for maybe one or two, maybe three substrates to bind to an enzyme. It would also allow for something like an inhibitor molecule to bind to the enzyme's active site and block substrates from binding. So the induced fit model allows for a couple of things to bind to the enzyme. However, the lock and key model requires only one thing binds to this enzyme, which is correct. Well, experiments using hexokinase help to solve this problem. Hexokinase is the enzyme that catalyzes the first step in glycolysis, where a glucose molecule is changed into a glucose 6-phosphate molecule by addition of a phosphate group. Kinase enzymes are the types of enzymes that do this, transfer phosphate groups from one molecule, so this ATP, to another one, so this glucose. Glucose is a near perfect match for the active site of hexokinase, but not entirely. It's not until after the substrate has already bound that you see the perfect shape match between these two. And so what happened was, I can't remember who performed this experiment, and you can find it here at this link here, but the person who performed this experiment did ball and stick model and 3D imaging of hexokinase before it binds glucose and while it is bound to glucose. And what they found was that there is a shape difference, an actual shape change between the molecule when it is unbound and when the molecule when it is bound. There's a change in charge, there's a change in shape. And so that showed that the induced fit model was actually the more accurate model for enzymes binding their substrates. In order to function properly and to orient their substrate in the active site so that it's a perfect, or so that the fit is good and that groups are arranged to help the reaction proceed, enzymes often require cofactors to catalyze that reaction. Cofactors are some type of inorganic ion, like a metal ion, or maybe something else more biological like a vitamin, a sugar, or a lipid that is bound to an enzyme that helps the enzyme catalyze a reaction. Without its cofactor, the enzyme is not functioning. Cofactors can be one of two things: inorganic metal ions, and that's where that one stops, or coenzymes. Coenzymes are more biological in nature. They have a biological source. Coenzymes can be cosubstrates or prosthetic groups. Cosubstrates are a second substrate of a multi-substrate enzyme. So an enzyme that has to bind maybe two things in order to catalyze a reaction: its substrate and its cosubstrate, some kind of cofactor. These are usually loosely bound and can come off the enzyme pretty easily. Prosthetic groups are tightly bound non-protein units required for function. So they still have a biological source. They can be vitamins, sugars, or lipids, but they typically are non-protein units. This is not correct. So please remove this from your slide deck, the inorganic metal ion. That is reserved for metal ions as cofactors. Prosthetic groups can be combinations of these inorganic molecules and some kind of biologically sourced molecule. So it can be some kind of combination of a vitamin with a metal ion, and that would be considered a prosthetic group. But metal ions alone are different types of cofactors. So what's the difference between a cosubstrate and a prosthetic group? Well, we talked about myoglobin and hemoglobin, right? And so the heme group embedded within a hemoglobin protein is a prosthetic group. It's a tightly bound porphyrin ring with a metal ion in the center. Hemoglobin does not work without its prosthetic group, right? That's where oxygen binds. And so if it's missing its prosthetic group, oxygen has no place to bind and it's not functional. Cosubstrates are a little bit different. And one example of a cosubstrate is NAD, which is represented here, NAD. NAD, or Nicotinamide adenine dinucleotide, which is why I don't call it that, we call it NAD, it's really hard to say, is a cosubstrate for many enzymes in biochemical reactions, specifically metabolic reactions. NAD is going to come back with force towards the second half of the semester when we start talking about glycolysis, the citric acid cycle, and so on. It's an incredibly important cosubstrate for enzymes, many enzymes. NAD's basis is actually niacin, which is a vitamin, a biologically sourced vitamin that you can get from your diet. It's modified by an amide group, and then the oxidized form of NAD is charged, and the reduced form of NAD has an extra proton and is not charged. This is what the molecule actually looks like. It's this full molecule here. And so it's two ribose sugars linked by a phosphate group, and then this ionizable amide-based group here. And so this is just one of the reactions that NAD can participate in. This is the end reaction here. And so if ethanol is to be converted into acetate by alcohol dehydrogenase, NAD must bind to the enzyme first, and then also ethanol has to bind. So those two have to bind together in order to catalyze the reaction, which is acetate, and then adding a hydrogen proton to this NAD. Cosubstrates have to be recycled, just like their enzyme. And so there's another enzyme that will oxidize NADH back to NAD+ to recycle this process. Many human diseases can actually be explained by deficiency in coenzyme. If you have a deficiency of niacin in your diet, that means you will not have the necessary base materials to make NAD. Without NAD, a lot of your metabolic enzymes are not functioning. And this can lead to, less NAD production. That's going to interrupt a lot of the redox reactions necessary for metabolizing sugar. And that's going to lead to a lot of problems, systemic-wide problems. Diarrhea, you're going to have a terrible diarrhea because you're not able to absorb the nutrients that you're consuming because the enzymes that process those nutrients are not functioning. You're going to have a lot of dermatitis where the cells that reproduce very, very frequently, like your skin cells, won't be able to do so because they're not going to be able to generate the ATP they need to replicate. Dementia will result because the necessary reactions that are required for firing synapses require NAD. And so without NAD, you'll have problems firing your synapses. And so just simply having not enough niacin in your diet will result in pretty widespread issues, because niacin is such a critical component of NAD, which is such a critical coenzyme for many different enzymes in biochemical reactions.